to do spectral imaging. If you start thinking about microscopy, there are different optical contrast methods that can be used. On this slide, I show kind of some of the more common ones, bright field and phase contrast at the cheaper, easier, older end of the spectrum. And as things evolved, dark field, polarization, and DIC, these things get a bit more complicated. Fluorescence is the most colorful. It's the most well known for its range of colors. And if you think about what fluorescence is, there might be some things that come to mind. If you were to ask maybe children's age, any of these images come to mind. But what I'm showing here are three images where really there's only one of fluorescence. Here we have phosphorescence, fluorescence, and luminescence. And when you're young, you use the words interchangeably, but because they all kind of mean chemicals releasing light. But what differs between them is how the energy gets in and how quickly it comes out. So on the left, we have phosphorescence, you probably know from watches and clocks. This is where the light goes in, but takes an extremely long time to come out. On the right, we have luminescence that you might be familiar in things like fireflies, where you don't need to put light in, just chemically, they have enough energy to release light. But fluorescence is what we care about in the middle, where we put light in, put energy in the form of light, and we get energy and light out immediately, very, very quickly. And why is that of interest? Well, it comes to all about the contrast methods. Of all the contrast methods, fluorescence has some really nice properties. The first is that it has very high contrast. So this means the signal to background. If you don't label something with fluorescence, it can be black, as you see in this hole in this kidney section here. Whereas when you do have something, it can be very bright. So you get brilliant kind of bright to dark state. Second thing is it can be very specific. So you can label just what you want to within the sample. And then the third thing would be that it's quantitative. What do I mean by this? Well, if you have a signal which is 100 gray levels and a signal which is half the intensity at 50 gray levels, in fluorescence microscopy, with some other caveats, you can be sure that that is linear and you have twice as much of something and half as much as something. Whereas in other techniques, like dark field microscopy, whilst it looks amazing, it can be extremely hard to impossible to quantify. And this really comes to when you think about what the word microscope means and what a microscope does. Microscopes really help you to see stuff. I know it sounds very basic, but to help you see things, you need three things. It's magnification, it's contrast, and it's resolution. So the magnifying part is the part most people think of to make it look bigger. Contrast depends on the technique, and it's what fluorescence gives us. And resolution, the ability to distinguish two objects which are close to each other. Now, in the early days of microscopy, once magnification had been organized, people began to work on improving the lenses and working on the second two parts of the equation, so the contrast and the resolution. And on the left here, I show a prototype microscope. This is an ultra microscope. This is a prototype microscope from 1902 for what became, as you may know now, the light sheet microscope. And the idea here was that you would excite light from the side and detect it orthogonally from the top. And by doing that, you can improve the contrast within the imaging system. Now, whilst working on the contrast on this prototype microscope, people began to think about the resolution side. And we worked for Carl Zeiss. One of our co-founders was a gentleman called Ernst Abbe, who this picture in the middle is his memorial in Jena. Now, one of the things he did extremely well was to work on the theory of optics, so turning it from the trial and error designs of the early 1800s into the scientific basis we have today. One of his most famous discoveries, hence why it's on the top of the memorial and enlarged at the bottom here, is the resolution equation, which fundamentally says how close can two objects get before you can no longer tell them apart. And up until the advent of super resolution microscopy, this pretty much held true for 100 years. And even today with super resolution, they build upon this. And in essence, what the equation says is there are two factors that affect resolution. The first is the wavelength of light you use, and the second one, the component at the bottom here, is effectively NA, the angle over which you can detect. So NA you can build upon and improve by building better optics, but at some point you hit a limit where you have as large an angle as you can have going into the sample, you're collecting as much light as possible, and you hit the resolution limit. And so what the gentleman who worked on the light sheet design on the left considered was, well, the other part of the equation is the top part, is the wavelength part. And so if we shift the illumination to lower and lower wavelengths, we can get better resolution. And in doing so, they built various designs. One is shown on the right here in 1908. This is a UV illuminator coming into the microscope. And what they discovered was not only was the resolution quite good, because it was a shorter wavelength, but that things glowed. Now this slide, I'll admit, is a little busy. I'm not going to read out all the names on this slide, but what I wanted to kind of talk through was some of the evolution that led us to the spectral systems we have today. And I will point out, Nature has an incredibly good series of pages I linked to at the top here, which talk you through the history and evolution. I only even include a subset on this slide. The general theme I wanted to get across, though, was that from the early days of Leeuwenhoek, there was a group of researchers who progressed the field of labeling and staining. So the ability to put contrast agents into your biological specimen in order to improve the contrast you see. And if you see the Myers work from 1986, H&E staining, that's still used in hospitals today because it's so effective. Secondary to that, there was a group of researchers who over time began to understand and revolutionize fluorescence. And the most, I guess the most famous of those in terms of fluorescence was George Stokes, who coined the term fluorescence and began to describe the essence of how it works. And I'll come back to his work later. And then thirdly, there's the steps that happened in terms of hardware evolution. And these three things kind of came together in the early 1900s with what I described. August Kohler and some of the researchers at Zeiss were working on these UV microscopes. They noticed that things fluoresce. The group that were working on fluorescence realized they had fluorescence things. And if you put fluorescence things under the UV microscope, you could excite them more efficiently. And then later on, people began to put filters in. And the other massive milestone was Johann Sebastian Plon, who invented the dichroic mirror and fundamentally the architecture that all fluorescence microscopes use today, which I'll come to later. But that invention in '67 really was the explosion of fluorescence microscopy and the time at which it became very widely used in the research settings. Now, just to understand kind of how things work in terms of fluorescence, here I have the electromagnetic spectrum in terms of what you've probably seen at school, and we care about the tiny part in the middle, the visible part of the spectrum. But what's significant here is the energy gradient depicted within the spectrum. So over here on the left, you tend to have higher energy, and on the right, lower energy. I always kind of think, over on the left, you have nuclear weapons. Over on the right, you have jazz radio. It's very definitely a high to low energy situation here. And George Stokes realized how that worked and began to describe it. And later on, the exact mechanism was understood in the terms of the Jablonski diagram. So it's a very kind of chemical, it's a very a detailed understanding of the electronics or the electrical state involved. But what happens is you put in light and that will excite an electron to a higher level. And then somehow it wishes to release that energy and come back to the ground state. And in doing so, there are different paths under which it may go. The one that we care about is the most common form, which would be fluorescence. So you excite, say with green light, it goes up to a higher energy state, and somehow it gets back to the ground state. Now, in that process, energy can be lost in other ways. And there's a bunch of ways here, but you can think of it as heat or other vibrations within the electron state. So it may kind of go via an in-between triplet state, and as it comes back to the ground state, it releases the energy in the form of an electron, and you see that energy. But because energy has been lost along the way, the photon that comes out is of a lower energy and therefore a longer wavelength. So typically, excite with green, see red; excite with UV, see blue. George Stokes described this phenomenon in the Stokes shift. So he said, if you excite at 550 nanometers, Alexa 555, you may see it at 570 nanometers. Now, there are different ways of describing this. The Stokes shift typically refers to the peak of these graphs. You may wonder why there is a distribution in these graphs. Well, the first curve, the excitation curve, is the probability of you exciting a dye. So what this means is for Alexa 555, if you use 550 nanometer light, you will excite it much better than using any other wavelength of light. If you were to use, let's say 515 nanometers, you would need to put twice as much energy in to excite it to the same level. If you use 450 nanometers, you might need 100 times as much energy. So the excitation curve, you want to try and hit your dyes closer to the peak if you can, in order to be a bit more efficient, but you can hit them anywhere on this curve. Then, depending on which way round the Jablonski diagram the electron goes before it's released, will depend upon quite how much it's shifted into the red. So if it's a more efficient route, it might be slightly greener. If it goes a longer route and is more energy wasteful, it might come out at a further red wavelength. And because this is happening in your fluorescent system, with millions and millions of photons, you get a probability curve. And that, in effect, is the emission curve. It's the probability curve of a photon coming out at any given wavelength. So if you fire in 100 photons and you were to measure exactly where they came out, you would see this kind of curve. But for simplistic terms, people just refer to the peaks. Then in a typical fluorescence microscope, you would have filters in order to try and separate those colors. And filters really come into three categories. There would be band pass filters, which would allow light of a certain wavelength to pass through. You get short pass filters, which allow light shorter than a certain wavelength to go through. Long pass filters, which would be light longer than a certain wavelength. So here on this graph on the left, you see kind of three typical filters here. Here we have a band pass, which would just allow a short band in the middle here, a long pass in the red, short pass in the blue. And you get different properties of filters, but most commonly people refer to what's known as the 50% cutoff. So if you have a filter that's described as a kind of 470 short pass, what that means is it's probably blocking down to very close to 470, maybe 460, and then it allows 50% of the light through at 470, and probably by 465, it's allowing most of the light to come through. In a typical fluorescence microscope, we would combine different versions of those. This is a GFP, or a fixed filter cube, as it's known originally. And in the microscope, you would have it in the cube orientation on the right. So on the right, not shown in the picture here, you would have a very bright light source, historically mercury. About 10 years ago, we started to shift with halide bulbs, now with LED bulbs. But you would have a white light source, in effect, or a broad spectrum source. It would then go through the filter marked as the excitation filter, which is shown as this, the red excitation filter on the left. So that would allow light to perhaps, say, 460-ish nanometers to go through. that would hit the excitation curve of fits. You then have a dichroic mirror at 45 degrees. That would, this is an upright microscope design shown here. So you would have a dichroic mirror that would reflect the light down, it would excite your sample, and then the light comes back and it goes through the dichroic. So you want the 460 nanometers of the excitation to bounce off that dichroic, but the 540 nanometers of emission to go through that dichroic. So that the dichroic is shown as this yellow curve here. And then finally, you would have your emission filter, which would just be what's shown as the white curve here, but would allow through your green light. And so these three filters act in tandem to effectively only excite this part in the blue, to reflect that blue down, to allow the green out, and to capture the green in your detection system. And that's how a typical fluorescence cube works. In the case of modern confocals, that might not work strictly with fluorescent cubes, and Calliope will get on to explaining that later, but fundamentally you're still sending the light of a certain band. And with one color, that works great. The problem comes when you introduce a second color. And the problem comes because, as I mentioned before, we want fluorescence because it's specific. But what happens when you've got two things there at the same time? So here we have the excitation curve of GFP. And now if you also have YFP in your sample at the same time, whilst they have different peaks of the excitation curve, the curves themselves overlap a little. So here we have 488 nanometer light, as we would use in a confocal, hitting GFP perfectly, but you also see it hits YFP to perhaps a third efficiency. So you would also be exciting your YFP here. Now, in the case of the emission, you also have a problem in that here you've got the GFP on the left and the YFP on the right, emission curves. And what you see here is there is some overlap. So it becomes very hard to pick a filter which purely captures the YFP emission here. And the result is, if you're capturing channels, you have GFP on the left, YFP shown in the middle, and then when you combine them in your merged overlay, you see a lot of false colocalization. You just see a yellow image. Now, if that problem is limited purely to the emission overlap and not to the excitation overlap, then you can image sequentially. And that very neatly solves the problem. So in the case of a confocal, we can do that frame-wise, where we scan across the sample using just one laser. So just a blue laser to excite our green fluorescence. And then we stop, we change over to our red laser, and we excite, and we get our further red fluorescence coming out, and then neatly separated. And we can, instead of doing that frame-wise, we might choose to do that line-wise, where we alternate line by line as we scan down the image. All confocals pretty much can do that these days. By doing it line-wise, you minimize the chance of your sample moving between acquiring the green and the red, and you get a nice clear image. The only minor downside is it will halve your acquisition speed because you are imaging sequentially. And you can also image line-wise bidirectional, so scan one line across green, one line across red. So if you do that, if you have dyes which have separate or clearly separate excitation profiles, overlapping emission profiles, you can go from the situation you see at the top, where you have co-excitation, to the situation you see at the bottom, where you have pure excitation, and you can separate the dyes out. And if you look at the merge diagram, at first glance, okay, one's yellow, one's green. Why does that really matter? Well, it matters because fluorescence microscopy is used for co-localization studies as much as anything, where you're saying, what's green, what's red, and where do the two coexist? Where do I see yellow? Now, if ever you do co-localization and analyze it using a scatter plot, these are the scatter plots you see. So for those of you who aren't familiar, you take all of the pixels in the image and you look at their green and red contribution, and you map them into the scatter plot shown on the left. So if it's dark in the green channel and dark in the red channel, you put it in the bottom left corner. If it's bright in red and dark in green, it goes top left, bright in green, dark in red, bottom right, and anything which is co-localized or overlap, we see in the top right corner. So in this case, these two structures are quite distinct. We don't see much co-localization, and you see kind of two pure lines along the axis. This is a nicely separated sample. If, however, they were excited at the same time, or the emissions captured at the same time, and there was heavy bleed through, you see what you see on the right. So in this sample, it's impossible for there to be a green pixel, which isn't also detected in the red channel, and you get this kind of distortion of your co-localization. So this is why you see yellow in your image. It's not really co-localization. It's not green and red in the same place. It's just green and the green signal being detected in the green and red channel at the same time. Now, in the 60s, 70s, 80s, in the early days of fluorescence microscopy was so basic that this problem could largely be ignored, because you could pick a bluish dye and you could pick a reddish dye, and your experiment was simple enough. You could do that. As time went on, labeling complexity got more and more complicated. So there were different dyes for different things, different ion sensitive dyes, voltage dyes. And finally, there hit a critical point in the late 90s, shown in the bottom left here with the work of Roger Chen. Now, the problem and discovery he made was GFP. So if I come back to the history slide here, what was fundamental was that in the 60s, 70s, we were all just using fluorescent dyes. FITS, fluorescene, which was discovered in 1871, was still the most commonly used dye up until 2001. So for over 100 years, it hadn't really changed. Eosin was quite commonly used. Not a lot had changed. And the reason it didn't change was it kind of worked, it labeled, it was bright, who cared? But people were beginning to realize that it wasn't specific. It wasn't so easy to get exactly where you wanted. And you could mislabel things. And there was all the issues of labeling. How do you get it into a living sample? And what the group of Chen, Shimura, and Shalif did, which led to them winning the Nobel Prize in, I think, 2008, was that they discovered that jellyfish have a green protein. They discovered this and found this in the 60s, and they'd worked on collecting it and synthesizing it. And the most significant point was in 95, Roger Chen realized that if you had certain mutants, one of them, the S65T mutant, was very, very bright. It had the excitation curve, which perfectly matched 488, and it had an emission curve, which was where fluorescene typically was. And what this meant is we had a protein you could put into a biological sample that when you put under your fluorescence microscope, which had been around for 30 years, perfectly matched the fluorescene filter set. So you had a bright dye, labs started using it all over the place. So in the following 5 years, GFP became very common instead of inherent labeling of samples. And then round about the millennium, people discovered what we call the XFP, so RFP, CFP, YFP, all the different varieties of fluorescent proteins and activatable proteins. And that led to a bunch of colors being used, and that led to spectral confocals. So on the bottom timeline, two things that came together were in 2001, confocals suddenly became very easily able to do FRAP. Yeah, there had been systems in the 90s, but 2001, pretty much every confocal could suddenly do FRAP, which meant you could bleach proteins and you could do interesting FRAP experiments and FRET experiments. And in 2002, Zeiss launched the 510 Meta, which was the first, what I considered to be a spectral confocal, in that it had a multi-array detector and unmixing. And really the Meta was the commercial answer to the problem raised by the XFPs. And what do I mean by that? Well, this graph shows the excitation and emission properties of the main fluorescent proteins that were coming out at the turn of the millennium. So most of you, I assume, will have heard of CFP, GFP, YFP, these variants. And these variants were discovered not just in jellyfish, but in other marine organisms. Predominantly, they were found in coral reefs and other things that were stuck living under the sea in the sun. And they were really useful for transgenics, but they also had extremely broad excitation and extremely broad emission profiles. Now, you think this is a bad thing, and from a microscopy perspective, yes, it is a bad thing. From the coral's perspective, it was a great thing, because if you're a coral and you're sat down in Australia in the sun all day and you're being hit by harmful UV and you can't have sun cream, what's quite useful is the ability to absorb the more damaging UV wavelengths and to do something with that energy, which in the case of the corals is to shift it across. So there's a quite well documented theory now that the corals and marine organisms developed this very broad excitation and emission curve as a way of dealing with energy that was hitting them. Anyway, that's an aside. The point was, labs all of a sudden had all these colors available in a transgenic form, but they had the problem that I highlighted about excitation and emission crosstalk. And you suddenly had samples where you couldn't use sequential imaging to get yourself out of a problem. And that's where the spectral confocals came in. And so in the early days, people would say, I need a spectral confocal. What do you mean? Well, over time, it became realized that when people say spectral confocal, or if you think right now about what do you mean by spectral confocal, there are different things, but in essence, there's three things people mean. The first is a tunable confocal. It means a system where if someone walks up and has, GFP was great, I can use my FITS filter, which is 30 years old. Okay, now I've got YFP. Do you have to go and buy a new filter, or can you just tell the hardware to detect a different part of the spectral range? So that's the first part when people say spectral. It means not having to go to the shops every time. Well, maybe not shops near you selling filters, but not have to buy a new system every time you use a different color. The second thing people meant was the ability to plot the emission graph. So the ability when you put GFP under it to say, can I see GFP? And that wasn't just because they didn't believe the papers that have been put out of what GFP looked like, but it was to see, what is my sample doing? Is my GFP really there? I think it's got GFP and CFP. Can I check that they're both there? Can I see what's going on? Can I look at, is there autofluorescence? So the ability to interrogate within my own microscope what that looks like. And then the third component of a spectral confocal is the last part, which is the ability to do something smart with that information to remove the bleed through. So in the bottom here, I show, this is an image of GFP, CFP, YFP shown as cyan, yellow, and green. So when you look down the microscope, it pretty much looks like what you see on the left, and you want to see it separated as you see on the right here. So at this point, I'm going to stop sharing and hand over to Calliope, who will explain how this these parts B and C come together and do the magic for you.
Mm-hmm. Okay, so I am going to continue sharing the presentation. I hope you can all hear me.
Yes, it's perfect.
Okay, so as Chris very nicely discussed, let me minimize this. Okay, so as Chris very nicely just discussed, spectral system has three key components. You can tune the detection wavelengths that you are detecting, you can plot an emission graph from it, and you can spectrally unmix the wavelengths that the emission spectra you're collecting. What does that really mean? So there are multiple ways in order to instigate this spectral separation. So these are three example systems that one could use. So the systems differ in first the way that they are detecting the system. So you can see that in the top two systems, you have a photomultiplier tube that is detecting one channel at a time, whereas in the bottom system, you have a multi-channel photomultiplier tube that can detect all the channels at the same time. The second point that they are different is how they are separating the light. So when you want to detect a spectrum, you need to somehow go from a single, essentially white light laser line to a dispersed spectrum. So in order to return the light into its constituent colors, you can use a diffraction grating or you can use a prism. So I think prisms everybody is familiar with. As the light enters the prism, it is separated into its constituent colors. And prisms are very useful. However, as they separate the light, they do not do that in equal intervals. On the other hand, diffraction grating is doing the same thing. It is separating the light into the constituent colors. However, it does so with very defined and equal steps along the whole spectrum. So, and a possible disadvantage of diffraction grating is the fact that it is slightly less efficient in dispersing the light compared to a prism. However, alternative methods can be instigated inside the microscopy system in order to collect all this extra light that might not get separated initially. So, why is my presentation not changing? Okay, I'm sorry, I'm having a computer issue. Okay. Oh, okay, now it's moving. All right. So as you can see here, we can have a close-up at how the inside of a spectral system in a Zeiss microscope looks like. So as you can see, the light is coming and separated already into the individual colors, and is projected onto a multi-channel photomultiplier tube that will detect the individual spectra. So this operation is done with a grating. As you can see here, the light hits the grating and then is separated. But what does that really mean? How is the light separation helping you in anything? So when the light gets separated and then essentially projected onto the detector, it allows you to collect what we call a lambda stack. So the difference of a lambda stack versus a normal image is that the lambda stack is essentially a three-dimensional data set. It's just the third dimension along with X and Y is lambda, so wavelength. So for every single pixel of your image in a lambda stack, what you have is that you have all the spectra that you mapped onto the detector. So it's not a single image, it's a collection of imagings. Therefore, it's called a stack, a lambda stack. So, so why is that very useful? As we discussed before, we have multiple fluorophores that are highly overlapping in their emission spectra. And therefore, you can imagine as we are separating the light, you can plot individual fluorophores effectively onto the different, essentially, parts of the lambda stack. So here we are looking at an actual stack that is taken from a polychaete worm that is labeled with multiple fluorophores. And as you can see, once we open up the lambda stack, we can see the different dimensions of it, and we know which wavelength its image is corresponding to. So what we can see is that we can use the wavelengths to plot, we can use the images to plot the wavelengths based on their intensity, and that can be used in order to further unmix the image. By unmix, I mean that we can use a software in order to identify all the individual spectra that were present in the images we collected and separate them spatially onto our image, going from a lambda stack that contains all spectra to individual images that contain the individual colors that we had in the sample. So here you can see we can separate the muscles, they're labeled in red, the central nervous system of the worm labeled in green, and the nuclei, I believe it's labeled with DAPI, so it's labeled in blue. So how are the mathematics of the unmixing working? So how does the software know what spectrum is what? So I'm not gonna bore you too much with the mathematics. This is a formula that you could use. And as you can see, we have the combined spectrum, which is what we are detecting when we are imaging with a lambda stack, and the combined spectrum is essentially the result of the addition of the intensity of the individual spectra summed up. So spectra have an additive property. So when you have two spectra at the same time, you can sum them up and get a greater curve. So in this equation, we can see that we don't really know the intensity of the die A and die B, so YFP and ZFP in this case. But this intensity information is what we can get from our lambda stack. So as you can see here, what the software does essentially is that it is trying to figure out how the individual spectra or of the dyes that we tell the software that are in our sample fit inside the combined spectrum it is detected. So as you can see, we have the GFP and the YFP, and essentially we're trying to calculate what is the proportion of each spectrum that is found inside the combined spectrum. So once the software manages to do the calculation effectively, it can give us the very accurate, a very accurate separation of the two or more individual spectra that were present in our sample. So as I said before, when we are summing up the spectra in order to, or when we are trying to unsum the spectra from the combined one to end up in the individual ones, the mixture is not always 50-50. So we can have different combinations or different contributions of the different dyes inside the combined spectrum. And as you can see, based on the different contributions we are having, the peak of the spectrum is slightly changing, as well as the amplitude and of the spectrum as well. So softwares are very able to do this separation and these mathematical calculations very efficiently. So how does the spectrum know what curves to use in order to pick out our proteins out of the combined spectrum? There are actually three ways to do it. So the first way is to use an interactive selection within single labels. So what does that mean is that you have some sort of basically reference dyes. So you load to the system, you load to the software some pre-selected spectra that you know they are the pure spectra of your protein. So you can either do that by labeling your sample individually with to have a control sample with each reference fluorescent protein, only one protein per sample, so that you can detect a clean spectrum and then load it and ask the computer to look for, ask the software to look for a specific spectrum. Or you can select by yourself. So for example, let's say you have a cell that the mitochondria are labeled with a specific dye and the cell membrane with a different one and the actin with a different one. You can by yourself place the area of interest where each of your dyes is present. Lastly, you can just ask the software to identify the spectra by itself by just telling it how many dyes have you added to your sample. So you can tell it to look for 2 or 3 or 4, and then the software will run some algorithms trying to estimate what fits best your sample. So of course, not all 3 strategies are equal. So in general, strategy 1, using reference labels, reference spectra of clean labels is the optimal choice. So it's very, it is something that you need to take into account before you start your experiment, so you can create the additional controls, but it is the best way to get the best results. So the second strategy is also good, like selecting by yourself what spectrum labels with structure, but it requires spectra that are nicely separated so that you can select by yourself accurately. And of course, it's more prone to user error. The last strategy is, I guess, the least optimal one. However, it is the easiest because you don't need any preparation, and softwares are getting better and better in automatic recognition. So you could try which one fits your experiment depending on your specific needs. So some things that can really influence negatively your spectral separation are illustrated in these images. So as you can see, There are several parameters that can influence how you detect your spectrum. So let's take the first strategy that you are imaging a reference spectrum, and then you're using this to unmix your images. If you change the laser lines you're using, so if you excite your reference spectrum with 488 laser, and then when you're doing your actual imaging, you excite it with 561, that's gonna change the curve you're getting because you're exciting the fluorophore more or less optimally. So you must keep the laser excitation the same between your control reference samples and your actual samples. Second thing you need to keep stable is the main beam splitters. So why is that? If you look on the right side of the image, labeled by the green 2, you can see how the main beam splitter can change the position where you get a dip in your curve. So these dips are normal, but they should be in the same position for every when you are trying to compare in between samples. Otherwise, you are gonna alter the way you're analyzing your results. So thirdly, you need to keep the same detection range because of course, if you don't detect the entire spectrum on both times and you detect different parts of the spectrum, you're not gonna get the same result. And finally, objectives have their optics. So they have an inherent ability to transmit light differentially in different parts of the spectrum. So it is important to use the same objective. Of course, you should choose an optimal objective for what you're trying to do and keep it the same in between the experiments. So in my experience, keeping all these parameters the same is sometimes hard to remember. So a great tip is to just use the reuse button. So in the Zen software, you can easily find there is a reuse button that will just copy all of your settings from your one experiment to the next. So you can make sure you don't change anything and nothing gets unnoticed. So the last thing you need to be careful about is overexposure. So we all know overexposure is not good, but when it comes to lambda scanning and unmixing, it can really influence negatively your results. So the reason is that when you are overexposing your sample, you are changing the shape of your curve. So if you look here highlighted by the red bubble, I guess, on the right-hand side, you can see that the peak gets elongated and more flat on the top. This can give the impression to the software that you have either additional dyes or that the proportion of the different dyes is different. So always check for overexposure of your samples. Another thing that you need to be looking at when you're trying to unmix your samples is the residual tunnel. So the residual tunnel is not exactly what one would think as the leftovers. The residual tunnel is the difference between how your references, so your reference spectra, fit theoretically versus how they actually fit in your experiment. So it's a difference between the experimental and the realistic fit of your references. What does that practically mean? Let's look at an image, we'll make it a lot more clear. So this is our polyketone worm sample. And as you can see, we have our different tunnels, the red, the green, the blue, and then we have the residual. So what you're looking for when you're checking your residual tunnels is, do I see any additional structures? So if you see any structures in your residual tunnel that you don't see in the other tunnels, that means that probably one dye is not unmixed. So potentially there is an additional dye in your sample, maybe an extra antibody or fluorescent probe was added and it wasn't informed in the beginning of the experiment. So it is important to see the residual looking as it does here, where you have a very simple, low background. So what is this background? This background is essentially what we call a shot noise. So shot noise is essentially photon uncertainty. When a detector detects, let's say, 100 photons, there is a certain amount of error that could happen. So this error is the square root of the number of photons you detected. So if you detect 100 photons, your error is 10. So you could have detected 90 or you could have detected 110. Software is not exactly sure, but that is a normal variation. On the other hand, when you are detecting 10 photons, of course, the square root of the shot noise is the square root of 10. So you have a much greater error. What does that mean is that the lower your sample is, the greater the soft noise you're going to get, the greater the noise you're going to get is in the end. So in this case, when you are unmixing, you're looking for a dim residual tunnel with no additional structure. Here you can see a residual tunnel that is generally fairly dim. However, you can see that the mitochondrial structures are labeled. That, for me, it would ring a bell that something is added to the sample, one spectrum is not unmixed, and then you should go back and ask, at least use the automatic finder and ask it to search for 3 components and see what you get out of it. So this is more or less the theory behind how we should organize and do lambda stack experiments and the things that you should take care of. So one tangential but very convenient thing that comes out of lambda scanning is that you can use the lambda stack acquired spectra in order to simplify your experiments in the future. So once you know what are the spectra of your protein, because you have already done a lambda stack and you have done your reference imaging and you know what you're working with, what you can do is that you can save the spectra that you have acquired, and then you can load them on your system and then ask the system to, once it's acquired the whole lambda stack, to select the specific slices of your lambda scan that are corresponding to the spectra that you're working with. This way you can essentially reduce the number of, the size of your data, because you are discarding all the non-relevant for this specific experiment lambda stack slices. However, it has the disadvantage that it does not allow you to go back and reprocess your data, because once the software acquires the lambda stack, it unmixes it automatically and discards the non-useful slices, the non-useful slides. So it is faster to analyze because it does it automatically, and the unmixing is easier, and you do get your results automatically. But if you change your mind and you think that your spectra originally were not very clean, you cannot change your data. So looking at some examples to see how all this theory works in practice. So here we can see actually a world record experiment where in 2006 we had 7 fluorophores. So these are very highly overlapping fluorophores, as you can see in the wavelength graph, and label individual structures inside the cell. And of course, back in 2006, that was the maximum number of fluorophores that somebody had tried at the time to add in a cell. So as you can see, lambda scanning and unmixing can give you really beautiful images. But of course, as Chris has mentioned in the beginning, times are changing, and they are changing very fast. We might get a new fluorescent protein per day, almost, if not more. So proteins are being modified again and again. The spectra slightly shift with every modification. And of course, we want more. The number of fluorophores per sample is increasing. So here you can see an example from later on where we are having 10 fluorophores, 10 fluorophores in one sample. So this is a sample of an intestine where essentially they are looking at the location. The researchers are looking at the localization of different immune cells in the gut, and that made it very important for them to be able to use multiple different fluorophores at the same time in order to see the interactions between the different immune cells. So another issue that is presented in multicolor or not so many colored imaging is autofluorescence. So we all know autofluorescence is more present in some samples than others. So for example, this is a plant. It does have very strong chlorophyll autofluorescence, and it's usually interfering with your experiment. So this sample is labeled with 4 dyes plus autofluorescence. And one thing that lambda scanning and spectral imaging is so good at is to separate the autofluorescence and remove it during unmixing, so it can allow you to visualize your sample. So when we look at the sample, this is a live sample, it's a plant, And we're looking at at the process of moving microtubules and intercellular transport. So what we can think is if we were to image these 4 fluorophores individually and then remove the autofluorescence, that would take very long. So you would need to image every fluorophore sequentially, and that would essentially make the studying of the intercellular transport almost impossible. So another example where we are finding the identification and removal of autofluorescence very useful is when we need to separate very weak fluorescence from autofluorescence. So one of the unfortunate side effects of autofluorescence is that we cannot really control how strong it is. So fairly often it might be even brighter than your actual fluorophores. So as you can see in this case, by removing the autofluorescence in magenta, you are able to actually see your sample. Once again, the sample is very sensitive or was very sensitive. And if you try to do it sequentially, you would blitz your sample. Another thing to remember is that unfortunately, autofluorescence is not only difficult to remove, but it is difficult to not get, because autofluorescence excitation spectra are very, very wide. So they can be excited with multiple laser lines. So that is also another issue. So in the last autofluorescence example, I would like to bring attention to the fact that not all autofluorescence is bad. So sometimes we like autofluorescence because it can give us some contrast, because it can give us some structural details without us having to label a sample specifically. So in this example of a liver fluke, we can see we have 2 types of autofluorescence. We have green autofluorescence and red autofluorescence, and we have DAPI labeled nuclei. So on the right-hand side image, you can only see the red autofluorescence and the DAPI. So the red autofluorescence allows you to visualize the structures in red, and in DAPI is your actual signal. So by using the spectra detected through spectral imaging, we can separate the different signals and use the autofluorescence that is useful for your experiments, discard the non-useful one, and allow you to move on in the best, fastest, most optimal way. So you all probably know about photoconversion. So photoconversion is, in this specific case with the Kaede fluorophore, is a very popular scientific method in order to essentially optically manipulate cells, change their color, and then allow you to study them further on and monitor processes such as migrations, etc. So here you can see we have created 2 regions of interest, one in the cell on the right and one in the cell on the left. And we can see how by essentially photoconverting the cell on the right, we are changing the actual wavelength of the emission of the protein, the fluorescent protein, allowing us to get the spectral separation, the different spectra from the spectral separation of photoconversion in these fluorophores in that specific case. So another example of how linear unmixing can be, and how unmixing and spectral imaging can be exceptionally useful for your experiments, is when you're using nonlinear imaging. So nonlinear imaging, optical imaging, or multiphoton or 2-photon imaging, it's all the same thing, is a technique in laser scanning microscopy where we are essentially using 2 photons to excite a single fluorophore. So if you remember the Jablonski diagram that Chris showed, when you hit a fluorophore with a photon of the right wavelength, you excite an electron that jumps up on the higher orbit, and then it goes down, and that creates fluorescence. You can also hit the fluorophore with 2 photons of half the wavelength, or of double the wavelength and half the energy, and have the exact same result. So one of the interesting things in 2-photon imaging or NLO imaging is that the spectra of the proteins are different when you're hitting them with 2 photons versus 1. And that allows you to basically activate multiple fluorophores with 1 excitation. So in this sample, we excited three fluorescent proteins with only one laser line at 780 nanometers. So this, of course, speeds up your imaging because you don't excite with 3 lasers sequentially. It does make it more gentle to the sample because you don't use 3 times the same amount of light on the sample. So you have less phototoxicity and less photobleaching. And with unmixing, you can separate your different spectra and you can get really beautiful results. Okay, so another type of imaging you can apply spectral imaging and unmixing is, of course, wide field imaging. So wide field technology is often considered as the most common, most normal, most widely used imaging technology. And here you can see how because you are acquiring, so in spectral imaging in wide field systems, you are exciting essentially the entire sample at the same time. There is no optical sectioning. That is the result of wide field imaging. However, when you are collecting all the light back and if you don't unmix, you can get really blurry images. When you apply unmixing in wide field imaging, you can very nicely separate the spectra of the different fluorophores that you imaged, and you can get much better results very fast. So another very interesting technology to use along with spectral unmixing is Aeriscan. So Aeriscan is a specific type of detector in the Zeiss LSM microscopes that allows you to get a significant 4 to 8 times actually boost in sensitivity, resolution, and signal to noise ratio. So when you are imaging with the Aeroskin detector, here you can look at the cell, and in this sample we have mitotracker red and we have Alexa fluoro 555. So mitotracker red and Alexa 555, we can see that they are significantly overlapping in their emission spectrum. So when we are looking at the image, in the combined image, we can see that we have a huge spectral overlap in between the 2 fluorophores. So by applying the unmixing algorithm to this data set, we can get much clearer images and very nice separation of the 2 spectra of the very highly overlapping fluorophores. And the nicest thing is that even though you are unmixing, you can still get exceptionally high resolution in your images. In this case, 140 nanometers. So you can use multicolor labels, but no actual compromise in the resolution of your images. So lastly, I would like to just highlight something I have already mentioned. So the nice thing about spectral imaging and unmixing is that it works for all technologies and it works for all samples. So you can use it with your wide field microscope, your multiphoton, your confocal, your bright field, everything. And you can use it for all of your samples. It's not something that is specific for a very specific preparation of a sample, for a very specific type of a sample. It can be used with most of your samples, most of your experiments, and can help you get better data in a very reliable way. So what I would like you to take as a take-home message from today is that a spectral system, as we discussed, is tunable, allows you to detect the wavelengths to change the wavelengths you are detecting in the software. So it doesn't pin you down to specific filters with specific emission and excitation range, or emission ranges mostly. It plots an emission graph for you. So that is essentially what I described before as a 3-dimensional stack, the lambda stack, that contains the XY information and the lambda dimension. So for every pixel of your image, you have all the spectrum in images, and allows you to spectrally unmix your dyes, autofluorescence, anything you have in your sample. It allows you to unmix it. So I think that if you would like to, that was the end of our presentation. If you would like to learn more about unmixing, please bring your questions forth in the Q&A session, and also don't hesitate to contact your local Zeiss representative or any of us for more information. So let me stop sharing.
Thank you very much, Calliope and Chris. If anybody have questions, please feel free to raise your hand and we will invite you to ask them.
I think there are some questions in the chat. No.
So I'm looking through the chat. I've had some come up and there are some general messages. The first one